as Hauke said, I'm really happy today to talk about the aquaculture, co-culture, a little bit multi-trophic aquaculture of macroalgae, and especially on sea grapes, which you can already see here, which is the algae I'm currently working on with for my PhD. And now in the first, let's say half of the talk I have approximately 25-ish slides, I think, so maybe half an hour I would take from your time to talk. And in the first half, I will talk a little bit about macroalgae and macroalgae aquaculture in general, and why I think it's really important and also for the future really nice, and what we do in our working group. And then in the second half, I will talk about the sea grapes and a little bit of the research I am doing. Yes, so what's the problem? Why should we do aquaculture? I will not go into this because I believe that here are lots of scientists and experts from the field, so I don't have to wrap up the problem again. But I think what we can say is that in the future, we might need to use the ocean in a hopefully very responsible and sustainable way. Currently, we have the planet Earth, which is covered by water by more than 70%. But based on data from 2019, we only receive approximately 2% of all our calories from the ocean. So there's definitely a potential to increase this number. And in my opportunity, I think algae are actually a very good idea. And why that is, we can look at here. I have figure from Duarte et al. These data are from 29, so a little bit old, but I think the idea is still the same. On the left side, we see the agriculture food chain with vegetables, with grains, grasses, and rice at the basis at a trophic position of approximately 1, which makes sense. And then we have some livestock like sheep or cows or other herbivorous animals on a trophic position of approximately 2. And then we have higher up in the food chain some carnivorous species, like in this example, it's bears and wolves. And then higher up in the food chain, even creatures that hopefully doesn't exist, like wolf eaters or the eaters of wolf eaters. So altogether, we can say that we have the agriculture, so on the lands, on a trophic position of approximately 1. And if we compare this to the seafood chain, we can see that on the basis we have the algae, we have seaweeds, phytoplankton, and then we have shellfish, like different mussels, or also the zooplankton, small fish, and then high up in the food chain, something like tuna and other carnivorous fish. So we have actually wild fisheries, which are on a trophic position of, in the mean average, 3 and more than 3. So compared to the agriculture food chain, way higher. And the mariculture actually is at approximately a trophic position of 2. So compared to the agriculture food chain, this is quite high. And I have a nice citation from the publication, which basically says that if we would reduce the trophic position, so of the mariculture we are talking about, for about 0.4, this would mean that we could double the production for the same use of the marine primary production. So basically, it means that if we start to culture organisms which are lower in the trophic food chain, we can overall have a higher production with the same input, basically. And now the question is, how could we do that? It sounds wonderful. And in our working group, we have several research ideas, which are probably not the only ones. But yeah, I would like to tell you what we are working on. And one thing is the integrated multi-trophic aquaculture. Maybe some of you are even here working with it. The basic idea is that we have fed aquaculture species. In this case, it's some finfish. It could also be a shrimp. We are also working with some carnivorous snails, which is basically fed. So we have a nutrient input, and then we have extractive species, for example, shellfish or sea cucumbers, which could extract the organic nutrients, so the particulated organic matter, but which can also be used by the farmers, so which would generate actually an additional income. And then we have an extractive species which could retrieve the inorganic nutrients. And here we could use a seaweed, or also some examples of halophytes. There was a PhD student in the working group of experimental aquaculture, Paula Senf. She worked on Zanzibar, and this is a publication of hers. She tried to design a low technology and also low initial investment recirculation aquaculture system using milkfish and sea cucumbers and some sea purslane, a halophyte. I didn't find a nice picture of a halophyte of this sea purslane. If you have one, maybe you can help me with that. And so this is a very nice publication if you're interested in it. And then there is another idea besides the IMTAs, which is the aquaculture of non-fat species. We are also working with this. There is, for example, this is the upside down jellyfish, which the postdoc in our group, Holger Kühnhold, is working with. The sea cucumber, we also do some research on that, as I already said. Mussels are, of course, also a really good example. And then there are the seaweeds, I am working with, especially with the sea grapes, as I said. Now, I basically hope I made my point clear why I think sea grapes are quite cool. And they can be used for a variety of applications, which could all basically meet multiple human needs. On one hand, we have bioactive molecules, which could be extracted for pharmaceutical uses. We have cosmetics. The seaweed can be used as fish feed, fertilizer, for biofuel, or even to produce bioplastics. I am especially interested in the use for the direct human consumption. And here, on one hand, we have the use for food additives like texturing agents, carrageenan, agar-agar, alginate, or as sea vegetables. So basically for the direct human consumption. And if we now look a little bit on the numbers, I don't know, maybe you can have a guess, because for me these numbers were impressive. How many of the produced global seaweeds are actually used for the consumption or as food additives? I will tell you, it's actually more than 80% of the global production of the globally more than 35 million tons, which were produced in 2020. So it's the overall average is basically used for food additives or for the direct human consumption. I actually first guessed that the other purposes are a little bit higher in the ranking. More than 95% of all of these algae are coming from aquaculture. And most of these aquaculture, more than 99%, is actually taking place nowadays in Asia. So I think there's a lot of potential in other regions of the world. I think in Tanzania, there's also quite a bit of aquaculture, but I think some of you are here, you might know that way better than I do, but I think there's a very high potential. Now I just want to show you this slide quickly, because 35 million tons fresh weight, for me, this number, it doesn't mean so much. It's very hard to imagine. So let's have a look at the Mariculture Production. These are not the newest numbers, so they are still up to 2018. I think now the new Sophia report of the FAO is out, so we would have newer numbers. I didn't update it yet. But what we can basically see is that seaweeds make up more than 50% of the global mariculture production, with finfish and crustaceans, including the shrimp, which a lot of people, I think about when we talk about aquaculture, having only like around 10% of the production. Now, the next question, I think, is, okay, we culture a lot of seaweeds, but what seaweeds do we actually culture? What species is it? And in order to do this, we will have a look at this figure, which I like a lot. It's from the Neophobia report from 2022. So we also have the numbers of 2020. It's a little bit, lots of numbers, maybe a bit confusing. So I would propose to have a look at the percentage of the total production. And what we can see is that we basically have three main species which are used for the direct human consumption, which is the laminaria, the Japanese kelp, which is known as kombu, which you can, I know it's eaten in soups and different dishes. Then we have the Unadaria pinnatifida, which is known as wakame, also brown algae, and the nori, the porphyra, which is it's actually, I think, a red algae, and it's known as these sheets, which, for example, sushi is wrapped in. So these are used for the direct human consumption. And then we have the Orchoma and Gracilaria seaweeds, including the Capaphycus everesi, and here is the spiny seaweed. So those are basically used for the production of carrageenan and agar-agar. And I think what, at least for me, is incredibly impressive, I would have expected it, is that these 8 major species make up more than 93% of the global seaweed production. So it's basically only 8 major species or groups. Green seaweeds are completely underrepresentative. They make up less than 1% of the global production. So this can actually, cause a lot of problems if we, for example, think about pests and diseases. I don't know if someone made experiences with it, but when we talk about the Orchoma or the Capaphycus seaweeds, I think ice is a very known problem. And if we have only single species or genuses that we are culturing, this means that farmers have huge problems and have only this one species. And if there is a pest or a disease happening, maybe also triggered by higher temperatures due to the climate change, then the whole harvest can basically be gone. So I think these are reasons why we could talk about diversification of the seaweed production. So maybe also culturing species which are locally abundant somewhere, which can be used for bioactive compounds. We know more than 10,000 seaweed species. So I think there's really a lot to do and a lot to explore. And this is also the reason basically why we started working with Chloroplast lentilifera, so the sea grapes or green caviar. The green caviar got its name, as you might see down here on the picture, from this very, very special texture. So it has little ramules, which are on this front. And if you basically eat it, it is filled with basically with salty water, and it reminds a little bit on the experience when you eat caviar. I must admit, I never ate caviar, but lots of people tried it and told me it is really like that. It's nutritional beneficial. It has lots of interesting proteins, unsaturated fatty acids, and minerals. And for me, especially interesting, high antioxidant activities, especially due to ascorbic acid and different polyphenols. Other functional compounds like zephanoxanthin or caulerpine might also be interesting. As we can see here, the sea grapes are actually often eaten fresh, which causes problems when we are talking about transportation or storage, because everything has to happen very fast from the harvest to the retail. So they can also be dehydrated, basically using a brine solution. And then in these little bags, they are coming in very high salty waters, and then they can be rehydrated using fresh water, only slightly salty waters. And like this, they can be durable 6 months to 2 years. That's at least what I saw. These algae are mostly cultured nowadays in tidal ponds in Vietnam, where we are working, and in the Philippines, but also in Thailand and some other places like Japan. The demand is, according to the literature, currently higher than the supply is. So with our farmers that we work with in Vietnam, actually most of the sea grapes are exported to Japan and sold there. Traditionally, the sea grape aquaculture comes from the Philippines. In the early 1950s, the farmer of milkfish were actually having some of these algae in their fish ponds, and then they figured out that they actually grow quite well in there. Traditionally, it is another species. It's not the Chloroplast lentilifera, but it's the Chloroplast racemosa, which you can see here on the picture. Both of them look quite similar. We will also look at some pictures in a little bit. And both are basically combined under the name of sea grape. Nowadays, the aquaculture is still happening quite a bit, probably also because the reproduction is comparable easy. The Chloroplast reproduce also sexually. However, in aquaculture, it's mostly done through fragmentation. So basically just a little piece of the algae can be taken, and from there you can really grow a full new population. This also can sometimes be a problem, especially if you ask, for example, people in the Mediterranean. There are like 2 species, I think, Chloroplast taxifolia and racemosa, which are highly invasive. They basically escaped from some aquariums, and now they're a huge ecological problem. So I think this fragmentation for aquaculture is quite promising. However, when we talk about invasive species, this really requires some caution. The culture can take place indoors. This is, for example, sometimes in Japan the case, or outdoors, which is, of course, way cheaper and actually also quite easy and with quite low initial investments compared to the indoor culture. Yes, I said already that we know the Chloroplast lentilifera, but also a lot of other Chloroplast species. We can here see the distribution, and more than 20 Chloroplast species are considered edible in a range of different countries. If you want to know more about it, there's a review from Subia et al. 2019 and de Galland in 2017, which are, I think, really nice. I know, first literature if you want to get into the topic. And there are at least 15 different varieties that are consumed in the Indo-Pacific alone. Here we can see a few species, the Chloroplast racemosa. It has a little bit bigger ramules compared to the lentilifera, but also some of the other species are edible. Actually, these racemosa, they are even, I'm not sure if I can say it, but I think they're even a little bit more tasty than the lentilifera. I did a little quick literature research, and I found a publication where they also found the Chloroplast racemosa in Kenya. So this might be really interesting to look into. And maybe there's more publications on this, that species also actually occur in this region. Yes, the work we do in the project Sea Grapes, and we are interested to do research basically along the whole value chain of the sea grapes in Vietnam, especially in Phan Phong Bay. And we work together with the Working Group of Experimental Aquaculture at ZMT, in cooperation with researchers from the Institute of Oceanography in Vietnam, Nha Trang, and with sea grape farmers from the farm Vija. So the most pictures you will see are actually from that sea grape farm. And we are interested in the aquaculture, in the post-harvest, and also in the retail. But I have to emphasize that I'm an ecologist, or I'm interested in the ecophysiology of the algae. But I think there's also a huge potential when we talk about social science questions. However, during my PhD, I only touched them very, very briefly, so I will not talk about this today. However, I think there's lots to do. Yeah, so these algae are cultured in these tidal ponds. This is the farm Vija. From the top here, we can see a picture from the pond itself. And I think the first thing that everybody recognizes is, why are all of these ponds covered? So basically, these algae are very, very sensitive to high irradiances, which is, of course, very nice for a tropical species. But I don't know if someone is working with algae, but then they will know we figured out that approximately 50 micromole photons per square meter per second of photosynthetically active radiation, so in short, PAR, are a good culture light irradiance. And if you work with algae, I think you will know that this is really quite low. So if we would, for example, measure the light outside on a sunny day in the air, we would measure in Vietnam values of 1,800 to 2,000 PAR. So these 50 PAR are quite low, and this is why the farmers do the effort to actually culture the whole ponds. The algae are cultured sometimes directly in the sediment. It's called the sowing method, or on these trays. You can see them here. So basically, the algae are just meshed between the two trays, and then the fronds start to grow out of the meshes. The farmers basically swim in the pond, and then they harvest only those fronds that have reached a certain size. And during the growth season, this can be done approximately all 2 weeks continuously. So here you can see a little overview. We have the tray culture or the sowing method in the tidal ponds. And then these algae can only grow in the Vietnamese or Central South Vietnamese dry period, which is from March to October. And the algae are harvested when they reach approximately 7 to 10 centimeters. They are sold fresh or dehydrated. And during the rainy season, the salinity will decrease and the temperature will decrease. And these algae can really not handle a lot of fresh water. So basically for overwintering, the parts of the algae are brought in tanks or indoors, or sometimes they are let in the pond. I will now talk a little bit about some research, or I have a typo here, about some research we are doing about aquaculture, and later I will briefly talk about some projects we are doing on the post-harvest. So I did not show you yet the location we are working at. This is the central south of Vietnam. Vietnam has a huge coastline of more than 3,000 kilometers with a lot of bays and lagoons. So there's a lot of potential for aquaculture. We work in Phan Phong Bay. And besides a lot of sea grape culture that's happening there, we have also the culture of Babylonia aurulata. That's a carnivorous snail, which is also of high value in Vietnam, and of Capophycus everetsi, which is also cultured around here. Why am I telling you this? Because we are currently working to implement co-cultures with both of these species. So I basically just returned a month ago from Vietnam, where after COVID, we were finally able to work in the field with the farmers and our partners. So I'm still very excited. And we did an experiment to look at the co-culture of sea grapes and Capophycus. Now the question is, why would we do it? And if we look in the literature, in theory, this is actually quite a good team up, because as I said, the sea grapes are very sensitive to high irradiances. However, the Capophycus is grown directly under the surface. So you might also know it from farms in new countries. I think there's also a lot of Capophycus, if I'm not mistaken. So it actually can grow under high light irradiances. So the idea is that we can actually shade the sea grapes with the Kappa ficus and maybe reduce the cover, which is costly and a lot of effort, this gather cover over the cultures. And then we have nutrient profiles that actually fit quite well together. The sea grapes prefer nitrate, whereas the Kappa ficus prefer rather ammonium. And then we have quite a high value of sea grapes, especially compared to Kappa ficus. So sea grapes can reach values of approximately $8 per kilo of fresh weight, which is quite a lot compared to the Kappa ficus. So we thought this might be nice for the farmers. Sea grapes are in Vietnam mostly cultured in ponds. However, there are examples where they are also cultured in open areas, so maybe in bays. This is, for example, a picture from French Polynesia. So this is why we were quite optimistic. And you can see it here. It looks a little bit wild because it was just after a storm. But we cultured Kappa ficus on monolines, and we attached sea grape cages, some with some gas material for protection and some not. And I didn't analyze the data yet, but I can say already that the ocean currents really impaired the sea grapes negatively. So I think planning to culture sea grapes outside of the ponds, really we are in need of a current protected area. However, we found a very high morphological plasticity, which is very typical for sea grapes. So they basically change their morphology a little bit according to the currents and other parameters. So this is something we are still working on. And then we are also interested in the co-culture with Babylonia arulata. My colleagues are still in Vietnam working on this. These are benthic snails, basically. They are most of the time in the sediment. And then there's a siphon, which they basically show out of the sediment. They are fed mostly with trash fish. So we have usually very high nutrient loads in the ponds. And the co-culture with sea grapes could basically bioremediate these nutrients and produce biomass. So also this is something we are working on. I think it's just a nice example because a lot of IMTAs are done with finfish or with shrimp, but actually we hope it also works with this snail. Also, of course, important is that these ponds of the snails are directly next to the ponds of the sea grapes. So I think it's actually, it could be applied because the retailers in the region are there. So this would not have to be implemented new. So this was me talking a little bit about aquaculture, and now we are also interested in the post-harvest. This will be a little bit more biological or biochemical. I hope stay with me. I just don't know what's your background. So I said that sea grapes are actually quite rich in antioxidants. So just a few words about antioxidants. Maybe you all already know that, but antioxidants can basically prevent oxidative stress, which is caused by reactive oxygen species, in short, ROS. And we can differentiate between enzymatic antioxidants, for example, superoxide dismutase or catalase, and non-enzymatic antioxidants, for example, polyphenols like tannins, flavonoids, or vitamin C and E. And these non-enzymatic antioxidants are nutritionally beneficial. So they actually play a role in protecting against certain diseases, like, for example, the metabolic syndrome. So it's actually a good thing to basically find them in the algae. And how do they basically get in the algae is a question we ask ourselves. And if we there look at the photosystem 2 and the reaction centers, we usually have light coming in order for the algae to do photosynthesis. And what happens is that we have electrons, which basically are handed down the electron transport chain and then end in the production of NADPH and ATP. And if now we have actually very high irradiances, So for sea grapes, these irradiances already start at 200 par basically. And this electron transport chain is saturated. So the electrons cannot be passed through the chain anymore. What's happening then is that the energy has to go somewhere. So we have a chlorophyll triplet state. And this is dangerous because this can cause the formation of reactive oxygen species, which basically starts a cascade of the production of other ROS. So as a response, the antioxidative system of the algae basically produces non-enzymatic antioxidants. So our theory or our research question is basically, could we use this, and can the antioxidant activity be triggered by the exposure to light stress? So could light stress actually be an inexpensive tool for sea grape farmers to increase the quality of the sea grapes? In order to figure this out, we did a few experiments. So just briefly, we basically had different irradiances from 50 to 600 micromole photons per square meter per second, the 50 par here being basically the control. Replicates of 5 over 14 days with artificial seawater and artificial light of 12 to 12 dark light rhythm. And then we measured the antioxidant activity and the total phenolic content. We used certain assays, which I think is not so important right now. And then we also thought that probably this will also have some negative implications, because these algae might get bleached. Light stress of 600 par, that's like useful for the algae. So we also measured the chlorophyll A content, and we analyzed the color based on the analysis of pictures of the different parts of the algae. So the front tip, the front base, and the stolon. So like this creeping part here. And I will now just very briefly show you what you found, because I think it's quite interesting. So we basically can see here over the time the antioxidant activity. And what we see is that it increases with the different irradiances. So up here, the yellow one, that's 600 par, the highest treatment we had, and it showed also the highest values. We can basically say that we were able to increase the antioxidant activity to more than 230% after 1 week at 600 par, and nearly 200% at medium irradiances of 200 par after 2 weeks. And in order to kind of get an idea how much this is, we also measured some fruits which are very high or known for very high antioxidant activities. We have here the initials, so basically the not stressed sea grapes, and we have here the light stressed sea grapes, which were stressed for 2 weeks at 200 par. We can see that they actually reached the levels of pomegranate. And yeah, the sea grapes triggered with more light for shorter periods even reached higher values. So I think this actually is a really interesting tool which farmers hopefully could use. But what we also found is that or that after the light stress, we actually saw bleaching in the algae. And we saw especially bleaching in these front parts, probably because these algae, I think I didn't mention that yet, are siphonous. So that means they are actually one large cell which has several nucleus. But it also means that the chloroplasts have the opportunity to avoid high light and to basically just wander in the parts of the algae which are not so much exposed to light. This is called chloroplast relocation. And this is, of course, a problem because we assume that color is a very important property for customers' choices. So, of course, here we have to basically find a balance between higher antioxidants and still a nice, appealing color. And what I then just want to say is that these picture analysis, we analyzed the red, green, blue color channels. And I was really excited about it. So if someone of you is working with algae, this might be interesting because it's a really cheap and non-invasive tool to estimate the chlorophyll A content. I'm just working on doing this with Kappa ficus as well. And there are codes of Winters et al., which can be used, which I think are really, really useful. So this was the more biochemical part of the presentation. And now I'm basically finished. Oh yeah, if you want to have more information, we just published a paper on this. And then there is one topic which I also like to talk and especially think about, which is that I think that seaweeds are a huge opportunity for the direct human consumption, also in places outside of Asia, where it actually is traditionally integrated. And here I would be very excited about your stories, because for me, as someone from basically Europe and Germany, I know that sea vegetables are only very rarely and locally part of the traditional recipes. So we have something, it's a halophyte, something called quella, which is sometimes traditionally eaten in northern Germany, but maybe at least comes close to eating algae. And if people are not used to eating new food, then there is a phenomena which is called food neophobia, which is basically the fear of trying new foods. And this is something which is very sad, because I think algae would be a really interesting food source. So I try to work with school children and basically just explain them about algae and make them excited. So for example, now during the winter times, we baked some algae cookies to have something which they might try at home to, basically, I don't know, just get the experience to try it out. I would be interested if in your countries algae are actually part of a traditional food, or if you think there is an opportunity to implement it. Yeah, and with this, I hope I didn't take too much into less time. I would say thank you very much. If you have any questions which we cannot, I don't know, talk about now, you feel free to send an email or I don't know, reach out on other ways.